What if you could make invisible microorganisms visible? Every bacterial colony begins as a single living cell. Let's discover how the pore plate method reveals and counts them.
Welcome to BioLens Learning, where science comes alive through every discovery. BioLens Learning. See, learn, grow.
First, collect the sample aseptically and mix it thoroughly before analysis. Prepare serial dilutions to reduce the number of microorganisms for accurate colony counting. Label each sterile Petri plate with the sample information before inoculation. Transfer 1 milliliter of the diluted sample into the sterile Petri plate.
Add sterile molten agar cooled to approximately 45 to 50 degrees Celsius.
Gently swirl the plate to evenly distribute microorganisms throughout the agar. Allow the agar to cool and solidify without disturbing the plate. Incubate the inverted plates under appropriate conditions to allow colonies to develop. Count colonies on plates containing 30 to 300 colonies and calculate the microbial concentration as colony-forming units per milliliter. Thank you for watching BioLens Learning. Stay curious, keep exploring, and never laboratory techniques. BioLens Learning. See, learn,